Welcome back to the Genesis Science Report. Tonight we're delving into a recent paleontological discovery where scientists were surprised to find collagen in dinosaur bones that are supposedly millions of years old. The problem is that this protein typically degrades over a very short period of time, and yet the claim is that it has persisted in these fossils for tens of millions of years. Now this raises some intriguing questions about the age of these samples and how their presence aligns with the historical record found in the biblical account. Joining us for this discussion is Joseph Hubbard, an international speaker, writer, and the director of Creation Research UK. Joe's work in paleobiochemistry provides valuable insight into how such discoveries can actually reshape our view on creation, biology, and even the age of the Earth. He oversees Creation Research's Global Museum Project and has been involved in academia for the last 10 years. Joe, it's great to have you back on the show.
It's good to be back with you. I'm looking forward to being able to get into this paper with you, but it's always a joy to be on the show.
I can't wait to dive into this. Listen, let's jump right in, because did researchers really find collagen in these ancient bones? I mean, shouldn't it have all been decayed after millions of years? And what methods do scientists use to identify this collagen?
Okay, well, this is very quickly becoming my area of expertise, right? Because this is what I've been studying for the past few years. First question, has collagen really been found? Collagen is a sort of a fibrous type protein. It's a soft, squidgy thing, right? It's a principal component of bone. We often think of bones as hard, brittle stuff, but they're full of soft, squidgy stuff as well. Well, have we really found it? I have a dinosaur bone here. In fact, I think I've brought this on the show before, and you can see we've drilled a hole in it. Now, that's been drilled deliberately, and we've actually even published on this bone now, 2 different publications, in fact, which I have here, in which we've identified collagen using mass spectrometry. Now, this is going a step further than grinding it up and putting it underneath a microscope, but with over 150 published reports now from secular scientific literature, we really do have conclusive evidence that collagen has been found. Now, you touched on this, David, because collagen is soft and squidgy, and these are what we call original biomolecules. They're originally belonging to the creature, they're still in the bone, and they should have decayed after just a few thousand years at the absolute most. So this problem of how do we get soft squidgy stuff left inside dinosaur bones is something that's really been challenging researchers for a long time, because it simply shouldn't be there if it's millions of years old. And of course, that's where this paper comes in. Now, you showed the press release, right? The press release made a few sweeping statements, right? Researchers have concluded that we've found the answer as to how this stuff actually survives. The original paper is a little bit more complex. You can actually see all the diagrams and all the squiggles. So we're not going to be able to get into too much detail in the time that we have, but in essence the argument is that they've been able to use some principles of organic chemistry, particularly the way that water bonds together, and they've argued that this explains how you can have soft squidgy stuff like collagen survive for millions of years. Now, I'm very quickly becoming a sort of expert in this field, but the one thing which I'm not an expert in yet is organic chemistry. So I make no bones about it. I've deferred to one of my colleagues from Canada, who's been a professor of organic chemistry. And when we first heard about this paper, we sent it to him for a few comments. And what's been interesting, Professor Ed Neeland is his name. You can look him up. He sent us a whole list of rather serious problems with the organic chemistry itself, as in the arguments that they're using is a little bit faulty. Let me just share one example with you. You see, the paper says this carbonyl oxygen has a pair of electrons that don't form bonds with any other atoms. Now, you may not understand the implication of the science there, right? It's a bit technical, but listen to what Professor Ed says. He says this is simply not true. Even isolating the molecule with the carbonyl group, like acetone for example, the electrons on the carbonyl oxygen resonate with the carbon atom, forming bonds. This is the very basic of organic chemistry. Now, you may not understand the terms there, but the point is they've got some very basic organic chemistry wrong, according to a professor of organic chemistry. So there's a number of issues with what they're actually trying to argue here, even on the face of this paper. It actually gets worse if you want to go a bit deeper. You see, the main researcher is quoted as saying, collagen is all triple helices from one end to the other. There's no weak link, and that's why I think it has survived. Now, you may pick up the I think there. You notice how they're not quite definite? Well, as Ed Neeland has basically said, 200 million years is a very long time, and I will guarantee you that hydrolysis will make mincemeat out of 99% of anything in that time, including all the rocks. In other words, from Ed Neeland's perspective, Professor of Organic Chemistry, it is ludicrous to suggest that any kind of organic chemistry can actually preserve these rocks. In fact, he concludes like this. He says, this is a rather poor attempt to use chemistry to buttress their faith system in deep time. I certainly wouldn't want to defend it publicly. So, yeah, there's a few issues with this paper, and it really doesn't actually answer this question of why are these things still here after millions of years? Mm-hmm. It's interesting when you think about it.
Yeah, it very much is interesting because even the abstract, even the public, the public published layman's level article, it says that this is mysterious. It uses the word mysterious. Mm-hmm. And it even admits, it says collagen's persistence is perplexing because the protein contains chemical bonds that should only last 500 years. And yet, then it goes on to say, somehow it lasted for millions and millions and millions. But this turning on this interaction to prevent hydrolysis or breaking down chemicals via water, you're saying that according to chemists, it's not strong enough to really make this perpetuate for tens of millions of years.
We're certainly dealing with a strong bond here, and there is some good science in there, but they've made some very basic errors with organic chemistry, but then they're arguing, even with their basic errors, they're still arguing for something that is impossible, right? There is always going to be a weak point, no matter how strong your bonds are, and over millions of years, it is simply going to break down, and you're still going to destroy it. In fact, the world expert on ancient collagen, a guy called Dr. Michael Buckley from the University of Manchester here in the UK, his quote is, there is less than 1% chance of less than 1% collagen remaining in less than 1 million years. Now, the letters are there to hedge his bets, right? But the reality is, given everything we know about the decay of collagen, the half-life of collagen, the shelf life of collagen, there should be no collagen in this bone after 1 million years. And yet here we have a bone that is supposedly, well, in this particular bone, it's supposed to be around 130 million years old. And yet it's full of collagen. And it's not just full of collagen, by the way, it's full of collagen that we can sequence. In other words, the collagen is in good enough quality to actually perform something called proteomics, which is not quite reading DNA, but it's reading the marks that DNA has left on the collagen, on the protein, right? So it's not just collagen or fragments of collagen, it's collagen that's good enough to be able to read. Now that's remarkable. We get archaeological stuff that isn't that well preserved. So it stands as a testimony to two things. Number one, this has been brilliantly preserved. It hasn't laid around for hundreds of thousands of years slowly waiting to be buried. It's been buried quickly, it's been buried rapidly, and it's been buried in a flood, no doubt about it. You can tell that by the rest of the fossils, that it's buried with seashells and the like, right? Drowned dinosaurs. Yeah. But secondly, no matter how hard you may try and argue using very complex organic chemistry, and no matter what sweeping statements the press release says, we still have not found an answer to how this collagen could last for millions of years. Everything within organic chemistry, and that was the point that Professor Ed made earlier, everything in organic chemistry tells us that time will simply destroy your collagen within just a few thousand years. So we still have very conclusive evidence that these bones are not millions of years old. They are in fact young. And of course, that fits quite nicely with the biblical history of events. A recent creation 6,000 to 10,000 years ago, and a global flood that buried all this stuff very quickly and very efficiently.
Well, now you and I were recently in Anaheim at the American Society of Mass Spectrometry, looking at liquid chromatography mass spectrometry.
Yeah. I was actually presenting this paper there, so
exactly. So you've done a tremendous amount of research in this, but collagen is not the only thing that is found in these samples, and certainly not the only thing that secular science reports are showing has been found. We're talking about osteocytes and proteins and even stretchy blood vessels in some circumstances.
Yeah.
Is that correct?
And nerve cells, and like, you know, we hear like osteocytes and stuff and blood cells and stuff, right? We're talking basically the soft squidgy parts of bone. Remember, bone is not just hard. There's soft squidgy stuff in there as well. And so bone cells are not just hard, you know, calcium structures like we might think of them, but they're actually living, you know, soft material. So just be wary, you know, soft tissue is a lovely layman's term, but soft tissue can also mean soft tissue that has turned to rock.
Yeah.
What we're dealing with here is not soft tissue remains as in preserved or calcified or anything like that. We're dealing with what we call original biomolecules, the original soft squidgy stuff preserved in the bone. The bone hasn't turned to stone. The bone has been permineralized, completely encased and encapsulated with minerals, and the soft squidgy stuff is still there. Now, this is really, really, truly significant, and I'll give you a little bit of a sneak peek into our next research bit, David. We've got the data, we're just writing it up now for secular publication, but we've been able to not only find a Jurassic crocodilian bone, which is supposedly 200 million years old, we have found collagen in it, we have sequenced the collagen in it, and we have matched that collagen in a database with 3 different modern day crocodiles. In other words, A, no evolution, B, soft tissue shouldn't be there, but C, well, this is evidence for what we call endogeneity. In other words, this crocodilian collagen inside this fossil crocodilian collagen, actually matches with modern day crocodiles. So that's really, really significant, and hopefully we'll have some publications on that out about that very shortly.
So Joe, this is basically, I mean, we're talking about, we're really close to actually being able to show sequencing of some of these things, and that's almost Jurassic Park's type, you know, what we think of when we think of the movies of Jurassic Park. But what it's showing is that crocodilian creatures in the past are extremely similar, identical in many ways to crocodiles that we have today.
Yeah, so there's no evolution at all, right? They're proteins. So we did this crocodile vertebrae sequencing. We then run the protein sequence. We got 41 protein sequences, if you're interested. We ran that through a protein database, and it came up with 4 matches. So the 4 matches were the American alligator, the Chinese alligator, the Australian saltwater crocodile, and the leopard gecko. In other words, 4 of the matches, out of the 4 matches, 3 of those matches were crocodilian, all of them were reptilian, so there's similarity within reptiles. But the fact that out of the 4 matches, 3 of them were crocodilian tells you that, okay, yes, they haven't evolved, they're very, very similar, and this is supposed to be 200 million years old, but that's not that unusual. We've known that crocodiles aren't supposed to evolve for 200 million years for a long while, right? But the significance is the fact that, well, this is a fossil bone from Peterborough in England. We don't have many crocodiles in Peterborough in England, right? And the equipment that we tested this on has never been tested using any reptile collagen before. In other words, the chance of contamination, or this being the result of contamination, is basically 0, right? It can't come from the wild, it can't come from the equipment, and so we're actually seeing really good evidence for endogeneity, the fact that this is indeed collagen from this bone. It's not contamination, it's not a bacterial microfilm or anything like that, because it's crocodilian for sure, and we've been able to sequence it and match it with crocodiles around the world. So stay tuned, hopefully that'll be published soon, but again, it's just more evidence that you can trust the Bible and that indeed the world is not millions of years old, and in fact you can go one step further because it's evidence that they haven't evolved either.
Incredible. I'm telling you what, breaking research is so exciting, and we want to thank you for being such an integral part of it. Joe, I cannot wait to see you again in person, and can't wait to have you back on the show to share more. All right, don't go anywhere. We will be right back with Viewer Voices after the break.